Hello, hello, hello. We are back with cell structures, plants versus animals, the showdown. All right, guys, our warm up today. The correct answer was three. Remember, nine is just bread, and one is not okay. All right, we have our agenda for the day. We started with what's under my microscope. We were looking at the importance of microscopes and how microscopes make things look so different. This, for example, is a stalk of celery. Stalk of celery. This is a cut of a spruce tree. Go ahead and Google those, they're beautiful. But this is essentially a cross section of a tree branch. Here we have a pine needle, just like a normal pine needle you pick up off the ground here in Georgia. This is a blood clot. We love these because these help us from not bleeding out when we get a tiny little cut or a nick on our hand. And lastly, this is an ant leg. This is an ant leg. And the reason we were looking at microscopic images and talking about microscopes was because we were talking about cells. And cells could not have been discovered without the invention of the microscope. So we are so excited for microscopic technology because now we can see the tiny, tiny, tiny way that things look and we can learn and discover new things. All right, guys, here we are. Let's talk about it. So did all these pictures look the same? So the answer immediately is no, they did not all look the same. No, they did not all look the same. And why? Well, we talked about the differences in color and shape and texture. And we discussed that they look different because they do different things. Remember, structure and function go together. So celery is going to have to absorb a lot and keep a lot more water than a pine needle. So see how these have these tiny little cells and these have these gigantic cells for water storage. Blood cells, for example, need to be super sticky and stick together, but that wouldn't be helpful to an ant's leg. He wouldn't be able to walk. So we are thankful to the microscope because we learned about cells, because now we can see cells, and we also learned that different structures have different functions. So we discussed that on the left side here is your actual cells inside your mouth, your cheek cells. Literally, we just take a little Q-tip, or excuse me, not a Q-tip, toothpick, scrape the inside of your mouth with a Q-tip. Make sure to wash your hands. We're going to wipe that on a slide and put a little bit of blue dye on it so we can see the outline of the cells. So here we see that these animal cells are more circular, while this plant cell is very boxy and very rectangular. These are all about the same shape, the same size, and they're all nice in a row. This is chaos. Just little circles, little ovals, all mixed together. And if we think about it, that makes a lot of sense because we need our cells to just be able to kind of moosh around and float in and out, where plants need to keep a really set structure so that when our doggos walk on the grass, it can bounce right back, or when wind blows it, it can bounce right back. It can keep its structure even when the outside elements are kind of coming at it. So that's super smart. Way to go, plants. All right, we did a little bit of a microscope simulation. I'm going to skip this for time's sake, but we talked about what do animal and plant cells look like. We just saw those. Are they alike? Yeah, absolutely. They're going to have a lot of things in common because we know, for one, they both have nucleuses. We know every cell has a cell membrane, so they have a lot in common, but they look different. So let's talk about why they look different. Now, this was our first independent graded assignment that's on accuracy. Everything else has been participation and just watching as we go and learning. This is a learning check. So, Miss H, oh no, I didn't do that. I zoned out. How do I get to it? No problem. I got you. You're going to go into our class modules. And right here, we love this handy dandy checklist. We love it. This is our favorite. So we're going to go to this handy dandy checklist. And right here, because it's Wednesday, class kick. First, second, fourth, sixth, seventh. I'm going to jump on first. And here's the thing. Well, Miss H, I started it in class, but I got distracted and I couldn't finish it. Do I have to start all over? No, don't you worry. Just put in the exact name you put in before, and it'll pull up the page that you had finished. So I already did the drag and drop. I already did the drag and drop here. I already answered my things here. All I needed to do was finish my 2 sentences, and it's here. So that's what I love about ClassGeek. Don't worry if you didn't finish in class and now you lost it all. It's right here. Just put in the exact same name you used. So I want to start over, so I'm going to go back to my link and I'm going to use a different name. So we're going to type out Miss Humphreys this time. Use a different name. And now I have a blank one that's come up. So we've talked about cell walls are the rigid outside structure. And remember, we think a wall, we think it's the walls of the building. You can't keep the building up without the walls. So our cell wall is this outside really thick structure. Okay? If you think about a cell wall like a wall, think about the membrane as the door. Okay? Remember, we compared the cell membrane to the bouncer. It is the ultimate vibe check. If something comes to that cell membrane, it's gotten through the cell wall, it's messing with the structure, we've now hit the cell membrane, it has to do a quick vibe check. Like, oh, you're good, you're healthy, okay, here you go. I don't like that vibe, you seem a little toxic, you kind of, it seems sus, you look like a bacteria, no thank you, bye-bye, okay? So, the cell wall is the outside wall, the cell membrane is the door, it's the bouncer, all right? Cytoplasm, I want you to think like plasma, like a gooey, not a nucleus, not a free-floating DNA, but like it's in the shoulders, okay? Cytoplasm is jello-like, jello-like. And that is what holds all those organelles in place, so everything isn't just sloshing around, bumping into each other. Now, chloroplast, this is rude because it did not go over, so we're just going to draw it. These little squiggly things right here are chloroplast. And chloroplast are what make plants green. Because humans aren't green, and we can't eat the sun, we don't have chloroplast. We don't need that bad boy. So that is a plant cell under 400 magnification. And remember, we learned about microscopes that the closer in you get, the higher the magnification, the more detailed. Again, love those microscopes. Thank you. All right, again, cytoplasm is the jello inside. The cell membrane is that outside bouncer. It's the vibe check. And we remember from our prokaryote versus eukaryote lesson that the nucleus holds the DNA. So it's got to be that dark spot in the middle. So that's my drag and drop for here. Super easy. And again, remember this is a human cheek cell. All right, this was done in class together. So remember, we had to add these text boxes again because ClassGeek. It says, type the organelles below and use the draw tool to X the correct columns. We're going to mark which ones are in which. Do not list the nucleus because all eukaryotes have a nucleus. So we know both Elidea and cheek cells, plants and animals, have a nucleus. So we're going to start with the cell membrane. Cell membrane, insane in the membrane. All right, now write this down, Snapchat it to yourself, text it to your best friend, write it on the bathroom wall. Every cell has a cell membrane, every single one. Bacteria, plants, animals, yeast, E. coli, kangaroos, octopi, mice, all of it, every single cell has a cell membrane. Every cell has to have a bouncer or it would be chaos. Okay, so both. Then we're going to go to, what was that other one? They were all started with C. Why do you think Miss Humphreys made us learn all the C ones in one day? Because those are going to be the really complicated ones because you're going to remember, oh, it started with the C. Well, there's still 4 more options. So we're going to start with these so we can get them down. Cell membrane. Cytoplasm. Sounded like jello. Ah, the jello, cytoplasm. Well, I got to have something to keep my organelles in place, and I know plants and animals have organelles. Now, so does bacteria. Remember, bacteria has its DNA just floating around. What's it floating around in? Cytoplasm. So, prokaryotes and eukaryotes have cytoplasm. All right, now let's talk about that green stuff. Chloroplast. Now let's talk about that green stuff. Chloroplast. Well, let me think back. Chloroplast is what makes plants green. Humans aren't green. Humans don't have. Animals don't have. Perfect. Plants have it. You say that thought process? Plants have chloroplast. Fantastic. Awesome, awesome. All right. Last one. Cell wall. Cell wall. And remember, I know that cell wall is what makes things rectangle so that I can bounce back when something knocks into me. But that wouldn't be very comfortable because if a human cells bounce back, oh, shit like this because you're a really rigid structure, that wouldn't be helpful to us, right? Our cells need to be able to flow through our veins, move in and out of our hearts and our stomachs. So cell walls are only in plants. Cell walls are only in plants. So your exit ticket today that you were asked to do on your own, remember we did this part in class, your exit ticket was to complete these 2 questions. What are some key ways the plant cells and animals are observed or similar? So you're going to look for those columns that are the same, that have 2 checks. And then it says, what are some ways that the plant and animal cells differ? Well, you're going to look at the ones that only have 1 check. And that was your exit ticket. Bye.